All right, it looks like it's 4 o'clock. Let's talk about lab reports. All right, so I want to talk about citations today. I'm hoping at this stage, most people have worked hard on your report and just putting on some finishing touches. So they are due tomorrow by, I think, 4:30 is the deadline, maybe 5 o'clock. I can't remember what I programmed it for, but hopefully everyone gets them in on time. I am available tomorrow during the lab time for consultation if you're looking for any last minute questions or touches on the formal report, but we are moving on with Lab 5 tomorrow. Today I want to talk about citations and just a reminder on how I want you to do them. I think most people did okay on the previous assignment in terms of the citations. There were just lots of little kind of tiny things where people were messing up. So just to clarify, you should have 2 citations in your formal report. One is going to be your scientific journal paper. And so make sure you include a copy of that file, that PDF, and it can be the same one that you chose for the lab, the pre-lab, or if you want to choose a different one, it doesn't really matter. What I'm looking for is some sort of sentence to show me that you can cite properly. I don't really care too much about what the sentence says. It should be kind of relevant to the lab, but mostly I'm just looking that you can cite properly and do it in the correct format. So you're going to do this. It's going to be in the introduction or the discussion, or both. If you feel the need, that's great. If you want to put more things and you want to cite the textbook and other things, that's fine. But I'm going to be looking for a minimum of 2 citations. The second citation is going to be the lab manual, and you're going to cite that in the methods section. So how to do a citation? It depends on the number of authors. They're usually done at the end of a sentence. So notice here, by the way, that the citation is at the end of the sentence and the period is over here after the citation, because that citation is part of the sentence, so the period doesn't go until the very end. Some people, for some reason, like to put the period here, and that is a no. Do not put it there. So one author is just the author's last name, comma, the year. 2 authors, the 2 names, so Smith and Jones is the example there, comma, followed by the year. If it's 3 authors, you're going to use this thing here, which is et al. That's Latin for et others, and that's going to be in italics. So al is abbreviated, so it has a period after it, and then a comma like before. Sometimes people like to cite within the sentence, and that is acceptable as well. And usually what you do is you're mentioning their name or names, and it's the same format. If there's 3 or more, it's et al. If it's 1, you just say Smith, and you're going to put in brackets would be the year. So the lab manual has no author that's been claimed on the cover or anything like that. It's many authors. I've written parts of it. People at University of Alberta, previous Canada employees. So it's kind of a bunch of people. And since it's given no listed author, you're going to use the college name. So the college name, of course, is Canada College, and then the year is 2021. So that's a citation. That's within the body of the report. The works cited list, this is the complete bibliographical information for those things that you cited. So this is the standard format for a journal article anyway, and this is directly out of Appendix A. If you want to look, there's more examples in Appendix A. You can see it's the last name 1, comma, initial, comma, last name 2, initial, and so on. Make sure you get all the punctuation in there. Some people like to put initials and no punctuation. Make sure all the punctuation is in there. The title is in sentence form, meaning that you start off with a capital and finish with a period, and everything is small letters in between. The exception is if you're using a proper name in there, it might have a capital letter. You're going to have the journal name, the volume number. The volume number should be in bold. I didn't put that, and then the pages. So that's a standard format. Here's an example of somebody I collaborated with, and you can see there's all their names, the year. Notice there's a species name right here, and it's in italics as usual. And the Canadian Journal of Microbiology, there is an abbreviation for that. You can either use the abbreviation or use the full name. There's the volume number, the page numbers. So take a look at Appendix A, because it shows you also how to do the lab manual and the textbook. There's examples there. So this is showing an example from Lab 2. And you can see the author there is Canada College. So one last thing to note is that in your works cited list, they should be in alphabetical order by the first author's last name. So in this case here, we have Stratting and we have Canna College. So that means in your list, Canna College goes first because K is before S. So let me know if you have any questions about that. Couple other general pieces of advice, and I think I've given this kind of advice before. Make sure you're specific and thorough, okay? You can see the examples there, right? The first sentence is, it's vague, it's not specific. The second sentence is very specific. So make sure when you discuss the data, you're discussing the data. You know, tell me the numbers, refer to them, talk about them. The other piece of feedback is, you know, this is a typed up, you know, you're trying to make it look professional and all those kind of things. So good language and grammar is a given. But sometimes the little things count. And, you know, you should learn how to use special characters. So carbon dioxide, you can see there's a subscript, right? Or micro for micromoles per liter. Figure out how to use these special symbols, okay? You guys are university students now. It's not very hard to do in Microsoft Word or in other software packages. And it looks so much better if you have the proper notations rather than, you know, I see a lot of people write, you know, H2O, for example, and it looks like this. How hard is it to make a little subscript 2? It's not. It looks so much better, and it's the right way to do things. So just a reminder of what your lab report is going to look like. Like I said, we're looking at maybe about 10, 12 pages, depending on how you end up formatting things. Each graph is its own page. Some people like to squish 2 graphs a page. I don't recommend it. You know, use lots of space. You know, we're not printing any paper off. We're not killing any trees that way. You know, make your graphs nice and big and use a full page for each of them. You can see what, these are the typical lengths of these sections. So introduction, 1 to 1.5 pages. 2 pages is okay. 3 is getting on too much and that kind of thing. Methods should be half a page really max. Results is about half a page. Discussion is about 3 pages and literature cited and so on. Appendix, some people are asking me about that. So your appendix includes your journal article, includes the rough data, so the tables, and also there's a couple calculations that, that were done for your tables. So make sure to include those. That should be easy marks for the appendix. Okay, so let's go back to the cytoskeleton, and we're going to talk about microtubules, and these are the 3 items of the cytoskeleton we're going to talk about today. So microtubules, microfilaments, and intermediate filaments. So let's go back to the microtubules, and these are the largest of the cytoskeleton, and we talked about them a little bit last day. If you remember, we were talking about that they were made out of tubulin. So anytime you see IN on the end of something, by the way, this is usually meaning some sort of protein. So you might be familiar with insulin. So insulin, right, has that IN on the end. It usually means some sort of protein, and these are tubulin, meaning they make tubes, and alpha and beta are the versions of it. So before I go any further, what I wanted to do was start a set of notes. It's going to look something like this, and we're going to fill it in as we go along. And you can see, I think I have something like this similar in your notes, and we'll come back to this, you know, after we cover a little bit more material. What was I going to say about this? We have actually started talking about microtubules, so I'm going to fill in that component. So microtubules are made out of alpha and beta tubulin. Okay, relative size. You don't need to know the exact sizes of these. These are the largest of the cytoskeletal components. If you really want to know, they are about 25 nanometers. But I don't care about the exact numbers. It's more that you know they are the largest. So I'm just gonna see if I can make this a little bit bigger. There we go. And the cellular location. These are found basically in the cytoplasm. And so you're going to see that a lot of the functions are related to things going on in the actual cytoplasm. And so all of these cytoskeletal components, by the way, you could talk about how they're important in cell shape. I don't really care about that. I care about the specific functions. So one specific function that we did talk about already were spindle fibers. So you can, by the way, you can write fibers either way. It's up to you. The British way is RES, and the American way is ERS, and we're Canadians, and you know, sometimes we don't know where we belong. I think I kind of like the British English a little bit better, but it really doesn't matter in terms of how you want to spell that. Okay, so I'm going to come back to this once we start filling out a few more things, and let's talk a little bit more about those spindle fibers. As I mentioned last day, I wanted to do some definitions for you. So back to the PowerPoint. Where's the PowerPoint? There it is. There's our centrosomes and our centrioles. And I mentioned last day I was going to define some of these things for you. So you probably remember centrosomes from your high school biology, and there's an image of them. And if you look carefully at these things, I think I mentioned last day that this is actually kind of little bundles of microtubules. And so you can see that's actually 3 there. And there's a bunch of triplets. So 1, 2, 3, 4, 5, 6, 7, 8. It looks like 9 triplets of microtubules. So a centriole, this here is one centriole. And a centrosome is 2 of them, and they're kind of perpendicular to one another. And the other term I was going to define was a centromere. So I have here, I want to write some definitions for these things for you. And just so that we know what they are, because these terms do pop up again and again over the next couple of units. All right, so a centrosome is something called an MTOC, which is really not as scary as it looks. That means a microtubule, organizing center. And these are found in animal cells. So plant cells and fungal cells, they have something else. Honestly, I can't even remember what that something else is. So you can kind of think of this as the originating, the organizing area where the microtubules are assembled together, and then they're going to spread out and form things like spindle fibers and whatnot. So one other thing to know about centrosomes is that a centrosome is 2 centrioles. And they are perpendicular to one another. So there's the symbol for perpendicular to one another. Just trying to squeeze everything onto one screen. Usually I have a nice big whiteboard space, and just trying to adapt here. So what is a centriole? You can think of a centriole more as just a bundle of microtubules. So I'm going to say that. And you can mention if you want that you've got 9 triplets, but I don't think every centriole is 9 triplets. So I'm just going to call them, we'll say that this is a structure made, I'll just call it bundles of microtubules. Bundles of microtubules. Okay, so one more definition. Okay, so a centrosome, centriole, and so what is a centromere? You might remember that from high school as well. And this is actually a piece on your chromosome. And so what is it doing? This is actually where the spindle fibers attach. So here's my definition. This is actually a DNA sequence. DNA sequence, so it's a specific chunk of DNA, and this is on a chromosome. So DNA sequence on a chromosome where spindle fibers bind. And remember that spindle fibers are just microtubules. So I'm going to put MT as a reminder where spindle fibers bind. Okay, so hopefully those definitions made some sense. Like I said, they're kind of all related with that cent in there. I don't know if that means center. I'm not really sure who named these things, but sometimes people get these names kind of mixed up. So microtubules. Microtubules are spindle fibers. What else do microtubules do? Microtubules are actually part of eukaryotic flagella and cilia. So this is something we're actually going to look at in tomorrow's lab, is flagella and cilia in eukaryotic protists. So flagella and cilia, by the way, are very similar structures. Flagella are just a lot larger. You can think of them as like tails, and so you can see there's the example there. That's a sperm cell, and it's like a big tail or a big whip used for motility. And you may remember that bacteria had flagella, but the bacteria flagellum rotated around and around and around. This one here does not rotate, it sort of flaps up and down like a big whip. Cilia are a little smaller, and they kind of flap back and forth in 3 dimensions. It's a little bit more like a figure-8 shape. And you can think of them as little tiny paddles or little tiny fingers. And they're pretty much doing the same thing. They're just helping it to swim, but they're tiny little versions of a flagellum. So I have some videos here. You can see there's on top, there's a sperm cell, and you can see that the whip, the flagellum, is kind of flapping back and forth and propelling through the medium. On the bottom, we have a paramecium. And if you look just around the surface of the paramecium, you can see those little hair-like fibers. Those are the cilia. And like I said, you can think of it as like, hundreds of little tiny paddles that are just flapping back and forth and allowing that thing to swim around. So let's talk a little bit about some of the other pieces of structure of these things. So these are the actual organisms we're looking at. We're looking at Euglena, which is a photosynthetic protist. And if you look carefully, you can see it has that flagellum on there. Basically flapping back and forth. Some organisms have more than one flagellum. I think Giardia is a good example of that, but Euglena have one. And here's the paramecium, and a little hard to see on this video, the cilia, but if you look kind of at its surface, you might see a little bit of a blur around the edge, and it's allowing this thing to sort of propel itself through the medium. So these are the 2 that we're going to be looking at in tomorrow's lab. I have several videos for you to view. So one thing to note is that flagella and cilia are not just things that are found on these unicellular protists. So I mentioned, sperm cells are obviously where flagella are found in animals. We also have cilia. So cilia, you can see here's our paramecium over here on the left, but cilia is also found in your windpipe cells. So all down your respiratory system, we have these cilia, and these cilia actually have a lot of mucus on them. And this protects our respiratory system. So, each day we breathe and we're bringing in air particles, and there's usually dust and sometimes microbes in there. And what these cilia do is rather than moving our cells, they're moving the mucus. And so any debris and particles and bacteria that go down our respiratory system, they get trapped in the mucus, and the cilia kind of push the mucus up and out. And we end up swallowing the mucus. Usually when you're not sick, you don't even notice. Right? It just sort of goes up and down your throat, and you swallow it. It's not a big deal. When you're sick, you generally make a lot more mucus, and that's when you start, sometimes you cough on it and gag on it and, spit it out, and it's pretty gross, but it's there to protect your respiratory system. So it's the same system, these cilia, by the way. You can see this cross section, it's a bunch of microtubules, and it's these little doublets of microtubules. We call it a 9+2 system. So I think I have another picture here of the 9+2 arrangement, and you can see, you can definitely see the. This is a cross section of some sort of cell in this 9+2 arrangement. Oops, skipped a slide there. This 9+2 arrangement over here. So I'm showing you on the right there a cartoon of the structure of a cilia or of a cilium or a flagellum. And one thing I want to point out is that it has a membrane around it. And so that's something I want to talk about a little bit about the structure. And you might want to grab yourself a piece of paper, because I'm going to draw you some pictures in a couple of minutes. Over the next few units, I'm actually going to draw quite a few pictures for you. And so it's always good to have a little extra paper. And if you like to do it in color, you can have different colors as well. So here's from our textbook. It's showing that 9+2 arrangement. And you can see all these little doublets of microtubules. And they're connected by these red things called dynein proteins. These are motor proteins. I'm going to talk about them in a moment. And this blue here is the membrane. So this is a very different structure from the prokaryotic flagella, which was not surrounded by a membrane at all. This is surrounded by a membrane because of these little red motor proteins. So there's a whole bunch of different types of motor proteins that are found in cells. And the thing you should know about motor proteins is that they move, and they use up ATP to do so. So I'll show you an image here that kind of tries to explain how these things move. So if you take a look, you've got these 2 doublets of microtubules here, And these motor proteins, they're kind of like little flexible, they're kind of almost like fingers. They sort of flex back and forth, or almost like feet in a sense. And you can see that they flex back and forth and allow the microtubules, the two bands, to sort of move back and forth. But the microtubule bands are sort of flexible, which you can see on the right. And so that ends up being giving that flapping back and forth kind of motion. And this all takes ATP, of course. So you can see on the bottom here, this is showing the wave-like motion of the cilia or the flagella moving back and forth. So by the way, there are all sorts of things in the cell that are reliant on motor proteins. Here's one more example. You can see this is a microtubule and we've got a motor protein, and this is actually showing vesicles moving along. So we're talking about these little vesicles moving across and around the cytoplasm. How do they move? They don't just float around by diffusion. They're actually going in specific directions, and they actually basically drive around on little microtubule tracks. And that's what's being shown here in this micrograph on the bottom left, and then this little video here on the right. So pretty cool things. So at this point, like I said, I want to draw some pictures for you. And so I'm going to get out my pen here and go to my virtual whiteboard. And what I want to do is compare the prokaryotic with the eukaryotic flagellum. So let me just pull up my whiteboard. Where is it now? I think that's not it. Where did it go? There it is. Is this my whiteboard? No, this is not the whiteboard. I started a whiteboard somewhere. Let me just, oh, there it is. I just got to figure out how to share it now. Okay, that's better. All right, so there's my whiteboard. And what I'm going to do is I'm going to draw a prokaryotic and a eukaryotic flagellum. And I want to kind of compare the structures and a few other things about these two things. So maybe what I will do is I'll start off with the prokaryotic flagellum that is on the left. And I'm going to draw a few things around it. So let me see here. What I'm going to do is maybe I'll start off with blue. It's already selected. So I'm going to start drawing small, and then I'm going to zoom in so that I can label this properly. So this is anchored in the membrane by a structure called a basal body. And so as I mentioned, it's anchored in the membrane. And so if you think about this, if this was a gram-negative, we would have an outer membrane. So there's my outer membrane. Outer membrane. And it will have a plasma membrane. And in the middle, we're going to have our peptidoglycan or cell wall. So there we go. Peptidoglycan like that. Okay, and then my flagella, it has these proteins that are assembled together. They're actually called hook proteins, and this structure here is called a hook. So if you think about it, if the flagellum was just straight and it whipped around, it's not going to do anything. So it needs to have a little bit of a bend to it. Okay, zoom out just a little bit. And then we have the big long part of the flagellum. So here we go. I'm going to just draw it out just like this. And it keeps going way past my diagram. And so this here is made out of proteins, and they're called flagellin proteins. So there's IN again, right? These are flagellin proteins. I'm not exactly sure actually if the whole thing has a name, whether that's called the tail or not. I just know it's made of flagellin proteins. Okay, so what else can we say about this prokaryotic flagellum that I've drawn? I'm going to just switch to a different color now. Maybe I will switch to, let's see here, how do I change my color? Oh, there we go. Pop that up. Let's try, oops, something nice and bright. So how about orange? There we go. So what can we say about this thing? This flagellum, it rotates. So maybe I meant, no, that'll work. So motion is that it rotates. And, you know, this is clockwise and counterclockwise. But it can go both directions, right? What else can we say about this? So this here, another thing we can say is how it's powered. So this is powered by something called the proton motive force. Proton motive force. So you're probably wondering, what is the proton motive force? Well, we have not talked about it yet. We're going to talk about it in a couple of units from now. So don't worry about it if you don't know what it is yet. But we are going to talk about it coming very soon. Maybe the last thing to say about this is the size. So how big are these things? So these are basically 1 to 5 micrometers. So bacterial cell is, 1 to 2 micrometers. So these are, roughly that size, sometimes a little bit bigger. Okay, so there is my beautiful prokaryotic flagellum. I hope you like that. And, like you can see, I am an amazing artist. And unfortunately, that's not really paying the bills now, is it? Okay, so over here, I'm going to zoom over to this other side, and we're going to draw a eukaryotic flagellum. So we're going to start off with a membrane as well. And these things are also anchored by something called the basal body, which is an entirely different structure. And I'm not even going to attempt to draw it. But what I'm going to do is draw my membrane. So the membrane sort of does this kind of thing. It sort of folds in on itself. And then there's my flagellum. So kind of looks something like that. So I'm going to do that again. Folds in on itself. And there's my flagellum. So what I'm trying to do right off the bat is show you that this thing is much, much bigger. So this, of course, is my plasma membrane. So I'm going to go in and just label that. Plasma membrane. Like I said, I'm not even going to attempt to draw the basal body. You can take a look on the PowerPoint if you're interested in what a basal body looks like. I know that's not the most amazing plasma membrane folding over on itself. Let me see if I can fix that. It's tighter. It's more like this. There we go. I don't know. Drawing is not easy. I have a lot of respect for artists who can do these things. Okay, so what else are we talking about here? Don't forget, we've got those microtubules. So I'm going to draw some microtubule doublets. And they're probably not going to be amazing, but here we go. There's one microtubule. There's another microtubule. And I'll draw one more doublet. Remember, there's a 9+2 pattern in here. But I'm just doing my best to be as neat and tidy as possible. Okay, so let's label that. So this is a microtubule doublet, we'll call it. Microtubule doublet. So what else are we talking about here? We also have those, remember we have those motor proteins, so we got to draw some of those. So that's why I'm getting this red pen out, and the motor proteins kind of look like this. Oops, what did I do there? I accidentally erased something. Let's try that again. So microtubules, or sorry, the motor proteins kind of look like this. They're a little Y-shaped, almost like a little leg. So I'll just draw them like this. Okay, all right, so maybe what I need to do is figure out how to make my pen thicker. Probably a way to do that, but I'm not going to figure that out now, but let me just draw a few more here. Okay, that's probably enough. So let's put a label on those. So these are dynein motor proteins. Okay, so let's make a few notes about this whole thing, just like we did with the last one. All right, so first note is what this is powered by, and we already mentioned that in the PowerPoint. This is powered by ATP. Okay. What else did we talk about? The motion Okay, where can I put the motion? I'll put the motion right over here. About. So the motion is back and forth. So motion back and forth. Okay, and what was the last thing? Size, I think. Where can we write that? I can write size. Maybe I'll write that down over here at the bottom. So size, and this is just kind of approximate size because you're always in biology going to find exceptions, smaller ones and bigger ones. But size on these things here is usually, I think there are some as small as 5 micrometers, but more like 10 micrometers. Oopsies, let's try that again. 10 micrometers to 300 micrometers. So some of these are massive. If a bacterial cell is like maximum, 3, 4, 5 micrometers, this is huge, right? On some of these cells. Kind of depends on the cell type in general. So the paramecium that we're going to look at, or sorry, not the paramecium, the euglena we're going to look at, I think, just trying to remember, these flagella are, I think maybe they're 30 micrometers or something like that on those. So anyway, there are my 2 types of flagellum. So just trying to emphasize that these things are very different beasts in terms of their structure, in terms of their motion, in terms of how they're powered. They just have the same name because they do the same thing, meaning that they help these things to swim, but the structures are very, very different. All right, so I hope you liked my diagrams. I'm going to move back, and just before I forget, I am going to fill in this table here, and I just want to add that we are another function for microtubules. So microtubules, we've got flagella, cilia, structure. And what else did we show? We also showed movement of vesicles, and other organelles sometimes move around as well, but vesicles are kind of the main one. So microtubules are very, very important. They help things move around. They help chromosomes move around. They help vesicles move around. And if you have a flagella or a cilia, they also help you as a cell move around wherever you're going. Okay, so there are other things that microtubules do, but those are kind of the big ones. So I wanted to show you lastly this video here. There's no sound on it, but what we're looking at is some sperm cells, and somebody has taken an instrument and they're going to cut the tail off. So think about this. If this was a prokaryotic flagellum, the tail would be cut off and that would be it. It would stop moving. But this is a eukaryotic flagellum, and so there's still ATP in there. And as long as there's still ATP within that membrane, it can still move. So you'll see that when I play this video, when they trim it off, the flagellum is still capable of movement, at least until the ATP is used up. So that was kind of a cool video. Okay, so we talked about the microtubules. Those were the biggest ones. Now we're going to talk about the smallest ones, and these are called microfilaments. So microfilaments are made out of actin. So let's go and just fill that in here before we forget. So I'm just gonna go, there we go. Okay, so microfilaments are made out of actin. And you can see, because of that, sometimes in some textbooks, we actually call them actin filaments. So I sometimes use those names interchangeably. I'm trying to be consistent with the textbook and all the language that I use, and the textbook consistently uses microfilaments, so I'm going to try to always do that. But I like the name actin filaments better because it reminds me that they are made out of actin. So these are the smallest, and like I said, again, it doesn't really matter that you know the actual how many nanometers, but for reference, they're 7 nanometers. And the cellular location, which you haven't talked about yet, is that they are near or beneath the plasma membrane. So they're kind of near the cell surface, just within the cytoplasm is where they are. All right, so back to the PowerPoint, and let's talk about what actin filaments can do, because they can do actually quite a few things. So notice the structure is different, right? I think, let me take a look, there's a structure there. So the microtubules were tubes. Actin filaments are more like a little rope, where you've got these 2 fibers that are twisted around each other like a rope. Notice they're talking about different types of actin. We've got F actin and G actin. It just means F means it's in the filament, G means it's free. I don't know, can't remember what the F and G actually stand for, but that's not really important here. Notice that ATP is involved in this. Of course, ATP is involved in so many things where you're building and taking things apart in cells. So it does say here, of course, that they are a structural network beneath the plasma membrane. So let's talk about what type of things happen near the membrane. It turns out quite a few things. So one area that we find microfilaments is in something called a microvillus or microvilli. So microvilli, microvillus. So what is that? You may have heard of these things. These are these little cells, and they've got these little structures here. So this here structure is a microvillus, and this is found in your intestine. And so they have this increased surface area to help you digest your food. And this is a specialized structure for a specialized type of cell. Think of it more surface area. And you can see that this is made out of both microfilaments and intermediate filaments. So it's part of that structure. This is near the membrane. So in order to change the shape of the membrane, microfilaments are usually involved. So microvilli, I'll fill out that table in a minute once I'm done talking about what actin can do in general, because it can do quite a few things. So for some reason, my Zoom control panel is kind of in my way. I'm just trying to figure out how to get it out of my way. Another place that you may have heard of actin is involved in muscle contraction. So if you take a look here, here's actin right here. Come on now. Here's actin, and it's forming these little strands, and in between is this other protein called myosin. So it turns out that myosin is a motor protein. And so what do motor proteins do? They consume ATP and they move. And so what you have are these two strands of things together. We've got actin here and we've got myosin here, and they move and slide in relation to one another. So I'll show you, I actually have a video here from a muscle cell, and you can actually watch and see the actin and myosin slide within one another. And so when they get closer, that's contracting a muscle, and when they get further away, they relax. So that's basically how muscle works. It's pretty cool. I think it's in chapter 50 or something like that in your textbook, or 52 or whatever. So you can see this is showing the relaxing and the contracting motions of actin and myosin. Like I said, I'm not going to cover this in any extensive detail, but just know it as an example of where we might find actin. And I guess know that myosin is a motor protein. So what else is going on near the cell surface? We also have this membrane that can change shape in amoeba. And we've talked a little bit about amoeba already, and you're going to get a chance to observe some videos of amoeba in this week's lab. So all this, what we have kind of near the surface of the membrane is this, this stuff called plasma gel, and that's loaded with actin proteins that can be disassembled and solidified to change the shape of the amoeba's membrane. And when it forms these projections, they're called pseudopods. Remember what a pseudopod is? This means a fake or a false feet. And this is how these things move around. So I have a video I'll show you of, this is an amoeba that actually I took this video. And you can see this amoeba is stretching out in this direction. And some of the videos you're going to look at in this week are actually videos that I took of some other amoeba, and even better videos in this one here. But this one I kind of like because it was moving really, really fast. Okay, so a couple more things that are going on. I'll go back to those notes. Something that can happen in plant cells is something called cytoplasmic streaming. Every once in a while, when we look at Elodea in the lab, I actually see this, and it's really, really cool. And what you're seeing is actually chloroplast moving around. So plant cells, you probably know, they like water, and diffusing water around is important. And so what they do is they just take their chloroplasts and kind of just move them around. And what this does is helps the water diffuse a little bit quicker. Okay, so one more thing to mention that actin can do. And I know I'm throwing a lot at you, but most of these things you've seen before somewhere. And you just got to think about things that are near the surface of the membrane. Is making a cleavage furrow. So we're going to talk about mitosis and meiosis. I think that's topic 12. So we're coming up to that, and we're going to talk about mitosis. And the last stage once all the chromosomes are done dividing, is cytokinesis, and that's the division of the plasma membrane. And in animal cells, the membranes pinch, and when they pinch, they form this cleavage furrow, and that's done by actin. Okay, so let us go back to our notes, and we'll fill some of those out. All right, so what were all these things we said? We talked about microvilli structure. My mind's already drawing a blank. Muscle contraction, cytoplasmic streaming, amoeba movement, that's actually called amoeboid movement. And what was that last one? I've already forgotten. A cleavage furrow. During cytokinesis. So by the way, amoeboid movement, there's another example of something that we can observe in protists that we also see in human cells. So we have various types of white blood cells that actually part of our immune system that move around like amoeba. They move around making pseudopods, and they're looking for bacteria to destroy by phagocytosis. And so we do have human cells that do that kind of thing, crawling all around our body in various parts and tissues. They're kind of creepy in some ways, but also very, very cool. Okay, so moving on. Lastly, we want to talk about intermediate filaments, and I'm hoping we'll have just enough time to do a Kahoot as well at the end. So if you want to load up the app, you can do that. So intermediate filaments are intermediate. That means they're in the middle in size. You can see 8 to 12 nanometers. So what are they made of? These have a little bit more variety to them in terms of what they're made of. They're usually made of what we call, I guess, keratin family proteins. And usually they can be very tissue specific. Like you have intermediate filaments in your axons and your nerves, and intermediate filaments in different, all sorts of different cell types. The one that's kind of common to most cells we've actually talked about already. This is the nuclear lamina. And so this is holding your nuclear membrane in shape. So like I said, there's lots of examples of these. That is the only example I expect you to know, is the nuclear lamina. That is the most important example to know. And if you take Biology 201, you're going to learn a lot more about these things. But in this class, that's the one example I want you to know. Sorry, two examples. The other example I already told you was this, the microvillus. So you can see the intermediate filaments are down here, part of that structure. So like I said, these are found different types in different tissues, and you have keratin family proteins. So there's our image of the nuclear lamina, that going back to topic 6, and this is found kind of just on the inside of the nuclear membrane. So kind of just in that surface there, and holding that whole thing together until it's disassembled during mitosis. That's the only time the nuclear lamina is not there. All right, so back to our notes, and then we'll do a Kahoot. So intermediate filaments, what were they made of? I told you they're made of keratin family proteins. And I'm also going to say the lamins, which is basically a subtype, which are part of the nuclear lamina proteins. So these are medium. And so what did we say? 8 to 12 nanometers. Cellular locations, we're just going to say the nucleus. And some of them are in specialized areas in different cell types. So the two examples I wanted you to know were nuclear lamina, and microvilli structure. Okay, so hopefully that is a decently comprehensive list. I know that this unit is just kind of a list of a bunch of things, but the cytoskeleton is very important. If you do more cell biology courses, you're going to hear lots about it. And we're going to talk about it in bits and pieces over the next couple of units. For example, microtubules and spindle fibers are very important. A lot of cancer treatments attack those kind of structures. All right, so I'm just going to go back to the PowerPoint for 1 second before I do the Kahoot, because I just wanted to mention the last part of this unit is that, what about bacteria? Up until recently, our textbook said there were no cytoskeletal components in bacteria, and that is false. Actually, we've known about this for a number of years now, that there are cytoskeletal-like proteins in bacteria. So if you take a look, this is a fluorescent stain of one, and what you're looking at is kind of a spiral shape. And there's 2 layers to it, and it's making that shell, it's giving that cell a rod shape. So this is actually basically a form of actin. You can see it has a dumb name. Some geneticists named it MREB. They like their codes, but it's basically an actin protein. There are microtubules involved in cell division in bacteria and so on. So kind of just the bottom line is to say that there are cytoskeletons in bacteria. They're just not quite as sophisticated because they're, of course, prokaryotic cells. Okay, so let's do that Kahoot, and that will finish off today. And I thought I had it loaded up here, so I'm just going to have to stop this share for a second and find Kahoot. So just bear with me for a second. So where is it now? Oh, there it is. Okay. This is the wrong tab. The Kahoot is over here. Okay, so here we go. It's loading and thinking. Okay, here we go. All right, there's the game pin, 4587055. All right, we're going to start this in 10 seconds. I think I have 8 questions for you today about the cytoskeleton. The green fibers in this picture are likely to be microtubules, microfilaments, or intermediate filaments. Hurry up, 10 seconds. Okay, that was a quick one. Let's show the media. This is in the middle of the cytoplasm. So these are microtubules. Remember, the edge is going to be microfilaments, and the blue stuff in the nucleus is going to be intermediate filaments. So the correct answer is microtubules, moving around vesicles and spindle fibers and things like that. Good job, Shreepy. Number 2, which filament works with the motor protein myosin to generate movement? Okay, a little bit better this time. Congratulations. So microfilaments are the same as actin filaments. And so this is, in fact, actin. Remember, actin is like a little 2 layers of rope wrapped around each other. And the other big part was the myosin. Question 3, which molecule is a dimer comprised of alpha and beta tubulin? Microtubules, microfilaments, intermediate filaments. All right, good job. So what was the question? We've got tubulin makes microtubules. Okay, we're getting there. Question 4, vesicles are transported along microtubules with the help of motor proteins. That is correct. Yeah, so motor proteins, the microtubules is the same structure as spindle fibers. So what's helping them is the motor proteins. Those are the things that hydrolyze ATP and they move things along. Okay, Michelle is coming in hot. Question 5, spindle fibers are comprised of, I just told you the answer, what spindle fibers are. So hopefully you were listening. That's right, microtubules. All right, couple more questions. Question 6, which is not a group of intermediate filaments? This is right from the notes we just did. Spindle fibers. Spindle fibers are not intermediate filaments, they are microtubules. All right, question 7 of 8. True or false? A eukaryotic flagellum rotates. Correct answer is false. Remember, this is like a sperm cell. It's going to flap up and down. It's not going to rotate. All right, last question. True or false? Actin filaments are responsible for a cleavage furrow during cytokinesis. True. So not too bad. A few wrong answers along the way. Let's see who has the best score. Levi gets the bronze. Michelle gets the silver. Who beat you, Michelle? Look what happened there. Oh, Tristan swung in at the end. Well done, well done. So if only I had money to give out as prizes. I'm sure that would make you guys super happy. So that's the end for today. We'll come back with topic 8 on Friday. So that's it for today. So I'll see you then.